Jekka. Welcome to MB Lectures. My name is Beck Carland and I'm the Senior Curator of the History of Collections. I'd like to begin by acknowledging the traditional owners of the lands where we work, the Wurundjeri and the Bunurong people of the Eastern Kulin Nation, and First Peoples language groups and communities across Victoria and Australia. I pay my deep respects to their elders past, present and emerging. And also to all the First Nations communities who so generously shared their knowledge and language with us during the development of Treasures of the Natural World. So welcome. This is the last lecture in the Treasures series, and we are so pleased to be live streaming tonight from the theatre at Melbourne Museum, albeit without a live audience. Tonight, I'm wearing two hats. I am the co-convener of the lecture series, so I will be your host, but as co-curator of the exhibition, I will also briefly introduce our interpretation intent for the exhibition and hopefully entice you all to buy tickets now that we're reopening. I'm also going to briefly present just one of my favourite treasures. There are many. And then I will introduce my colleagues, Dr. Robin Wilson, Sarah Babista, and Hayley Webster, who will dive a little more deeply into their favourites. This exhibition features over 200 important objects from the Natural History Museum in London. The exhibition charts a course through seminal moments that changed Western science and shaped our very understanding of the natural world. Museums Victoria and the Natural History Museum in London have much in common. We both care for extraordinary collections and support world-leading research. And for both institutions, it is our exhibitions that provide the moment of connection between the stories we hold and wider society. When we began thinking about how to stage the exhibition here in Melbourne, we were privileged to be guided by the Yulenge Expert Advisory Group. Yulenge is a Koori word for knowledge, and the committee is made up of elders from across Victoria. We worked closely with Wurundjeri Elder Ani Dai Kerr and Bunurong Elder Ani Carolyn Briggs to respectfully position the exhibition here on Kulin country. But in particular, we worked with Barak Barap Yorta Yorta Wemba Elder Auntie Esther Kirby, you can see here in the red on the right. Also, Auntie Justice Nelson from Jajawurrung and the amazing Uncle Larry Walsh of the Tanarong Nation. And that's Uncle Larry on the left with the denim jacket on, whose beautiful words actually close the exhibition. Together, the elders emphasised the interconnectedness of humanity with the world around us. And they encouraged us to see these objects and specimens as treasured guests that travel the world to share their vital stories about life on Earth. As you enter the exhibition, you will hear a beautiful spoken piece which introduces Bunjil's law and provides this Koori perspective on how to consider the exhibition. The elders also asked us to reach out to some of the First Nations communities these objects came from and to seek permission to tell their stories and use their language names for animals and places. That collaboration has influenced so much of our approach, including the international communities we've collaborated with, the hierarchy of information in didactic panels, and the design of the exhibition, and even the materials we've used. The exhibition is constructed from 2,000 recycled boxes, which at the end of the exhibition will be composted. This is a first for us, and I think you'll agree the results are spectacular and impactful. Along the way, we hope visitors will be inspired to protect the greatest treasure of all, the natural world itself. We hope the exhibition is a reminder that we are not outside of nature. We never were. We are a part of the great web of life, a small part, but a disproportionately powerful one. So now to meet some of the extraordinary treasures that you'll find in the exhibition. I'm going to kick us off with not the biggest or the oldest. In fact, in comparison to some of the more spectacular objects in the exhibition, one might walk right past this one. But for me, it represents the richness that comes from First Nations collaboration, and it's become one of my firm favourites. This tatsao, or copper nugget, has historically been treasured or interpreted at the Natural History Museum in London since 1771 as evidence of the grit and determination of British explorer Samuel Hearne, who retrieved the nugget from treacherous terrain 400 kilometres south of the Arctic Circle. He was the first European to make the overland excursion across northern Canada to the Arctic Ocean. It has been unchallenged as an heroic story of exploration and the discovery of the northernmost copper deposit in the world. However, as I mentioned, the ULENG Knowledge Committee asked us to consider the colonial lens through which these objects and their collecting events have been understood. They asked us to reach out and seek First Nations perspectives. And when we did, a much richer, nuanced story emerged where the knowledge of First Nations communities became central to the narrative. And their agency and ongoing stewardship of the country reveals a very different story. This is Fred Sangress, Chief of the Yellowknives Dene, whom we were privileged to collaborate with on the interpretation for this object. Willede Yellowknives Dene are one of the peoples of the Tatsodine Nation, and their country is called Akecho, in the north of what we call Canada. Fred shared with us that the Yellowknives Dene have collected copper from the lands they call wide and open for thousands of years. It is a sacred resource for making tools. In fact, the people were known for pots, knives, and other tools that were made from copper collected in the northern part of their territory. Tatsao means copper or metal, thus the name Yellowknives was the knives they were holding in their hands made from copper. As soon as we started working with Fred, Hearne's individual exploits quickly became less interesting within a narrative of continual care for country over thousands of years. Fred also revealed that Hearne had relied on Yellowknives Dene on the expedition to find the copper and then to return with the nugget. When Hearne had set off to chase rumours of copper, he failed to navigate the territory twice. And on the third attempt, Yellowknives Dene joined the expedition as hunters, cooks, and carriers. Whilst they assisted Hearne through the rough terrain, the true focus of these traditional custodians of the land was to keep the most precious copper deposit secret, which they did. Ultimately, Hearne found little more than this 2.95 kilogram nugget on display in Treasures. Today, the Yellowknives Dene continue to thrive in their communities of Ndilo and Deta in northern Canada. They continue to campaign to protect their country and resist the ongoing impact of colonisation. With the knowledge and history of the Tatsodine Yellowknives reinserted, the nugget story becomes one of sovereignty and resistance. Fred assured us the copper fields remain a sacred resource for making tools and a true treasure of the natural world, and definitely one of my favourites. To our collaborator, Fred Sangress, we say, Masi Chow, thank you. Sarah Babista is an objects conservator at Museums Victoria. She works closely with collection managers, curators, and exhibition staff in facilitating access to the museum's vast collection through exhibition and loan programs. She is passionate about the role conservation can play in exhibition development and has a long-term interest in implementing innovative preservation strategies for collections on display. Sarah is the project conservator on Treasures of the Natural World, and tonight she will share the object that has most held her interest during that role. I'll hand you over to Sarah.
Thank you, Beck. I would also like to begin by acknowledging the traditional owners of the land where we work, the Wurundjeri and Boonwurrung peoples of the Eastern Kulin Nation, and First Peoples language groups and communities across Victoria and Australia. I pay my deep respects to elders past, present, and emerging. As Beck mentioned, this evening I'll be talking about my favourite object in the Treasures exhibition, which is the preserved skin of the Ice Age giant ground sloth. I'm not a scientist, I'm a conservator, and one of my primary focuses is implementing conservation strategies which are aimed at slowing down the deterioration of objects. So I was immediately intrigued at how a piece of 13,000-year-old skin, which would have normally decayed, could survive in such good condition and provide us with unique insights into these now extinct animals. So here's the piece of skin on display in the exhibition. It measures about 45 by 60 centimetres in size, so it's quite a large piece. And as you can see from the images, it's in remarkable condition, with the sloth's long brown top hair still present and the preserved thick epidermis or outer layer of skin visible on the underside. The skin was found in 1902 in the Mylodon cave, which is located at the southernmost tip of the Patagonia region in Chile. And its discovery, along with a piece of fossilized dung and bone, which are also on display in the exhibition, took place 7 years after the first Mylodon finds were made by the German explorer Hermann Eberhard, who, upon visiting the cave in 1895, came across a large piece of skin with hair. And here's the skin that Eberhard found, which is actually also in the collection of the Natural History Museum in London. The nature and unusual preservation of the skins, coupled with accounts of sightings from the Tahihosh, the first peoples of the area, led some to believe that they actually belonged to a recently dead animal rather than an extinct species. And this caused much speculation and comment in the scientific world and sparked expeditions to the area in search of a living sloth. Thanks to radiocarbon dating, we now know that the skin belonged to an animal which died 13,000 years ago. So since these discoveries, much has actually been learnt about giant ground sloth, with over 100 genera now recognised and occurring, though not all at once, over a period of about 35 million years, starting first in South America before migrating into North America about 8 million years ago. And some examples are shown here in this diagram. In the Treasures exhibition, the material on display belongs to the species Mylodon darwinii, which populated South America and lived during the Pleistocene epoch between 1.8 million and 12,000 years ago. The first fossil, which you can actually see here in the image, was found by Charles Darwin, the famed naturalist, who discovered this jawbone in 1832 in Argentina on the expedition of the Beagle, which spent 3 years surveying the South American coast. On finding the specimen, Darwin identified it as belonging to the same family as modern sloths. However, in 1840, Richard Owen, the renowned paleontologist and anatomist and the first superintendent of the Natural History Museum, described it as a new species and named it in honour of its discoverer. So since then, skeletal remains of Mylodon have been found throughout South America, from Bolivia in the north to southern Patagonia, showing that they were adapted to life in arid and semi-arid savannas, warm and humid forests, as well as cold environments, such as mountain slopes. They are estimated to have weighed between 1,000 and 2,000 kilograms and been about 3 to 4 meters long, with a short neck, powerful chest, sturdy, blunt snout, large jaws, and a wide, muscular tail. And although it was primarily a quadruped, its trackways show it was actually capable of walking on 2 feet. And surviving claw sheaths reveal that they had 3 wide, flattened claws on each paw, which are thought to have been used for digging up roots and tubers, stripping branches, and also defending themselves. Mylodon and its relatives are distinguished from other ground sloths by the presence of upper canine teeth, triangular cheek teeth or molars, and a small first toe on their hind limbs. And although abundant postcranial material has been discovered, at present much remains fragmentary, and a complete skeleton has never been mounted. Consequently, reconstructions like the one you see here in the image remain largely speculative and based on closely related species. So, like modern day sloths, giant ground sloths were vegetarian, and they ate a wide variety of plants, including grasses and sedges, as well as the foliage of trees and shrubs, which has been determined based on their dental characteristics, jaw biomechanics, and analysis of their preserved dung or coprolites, an example of which is on display in the exhibition, and you can see in this image here. However, new scientific discoveries are always being made, and recently, chemical analysis of amino acids preserved in ancient sloth hair has provided evidence that they may have, in fact, been omnivore, at times eating meat, most likely scavenged to supplement their plant based diet. And researchers have theorized that this consumption of meat was most likely for energy or to increase their metabolism in order to maintain a constant body temperature in the icy and cool conditions in which they lived. A particularly interesting feature of Mylodon is the presence of dermal ossicles, which are small mineralized structures consisting of calcium phosphate and collagen, which are embedded in the skin under the fur, some of which can actually be seen in the skin that's on display. Found frequently in reptiles, ossicles are extremely rare in mammals, with armadillos being the only living mammal to have the same protective feature. And here they are in much larger numbers on another piece of preserved skin and enhanced in this very early X-ray from 1906. Ossicles were located across the shoulders, neck, and back, and they functioned like a coat of armor, adding an extra layer of protection to the thick skin and coarse hair covering the sloth's body. Interestingly, the small number of ossicles present in the skin on display suggests it probably came from a section of the sloth's body, such as the belly or perhaps an armpit, where this protective rigid structure wasn't required. So, how did this amazing material survive? Well, we have the perfect preservation conditions of the cave to thank for that. At around 200 metres deep, 121 metres wide, and 21 metres high, the cave maintains a constant cool temperature and stable relative humidity because its size means that it's thermally insulated from the external environment. And so, in these cool, stable conditions, skin can undergo a natural freeze drying process. And once dry, it can be preserved for a really long time. Inside the cave, the skin was also protected from the harmful effects of UV light, which can break chemical bonds, accelerating deterioration. And of course, it was also protected from water, which would have led to damage and decay if it had become wet. Skin is made of collagen and protein, and this is a food source for some insect species. Just think of your woolen jumpers at home. At the museum, we protect similar material by periodically freezing objects to keep any hidden bugs in all stages of their life cycles. In the cave's cold conditions, pest activity would have been limited, and the carcass would have also been protected from other vertebrate scavengers, which may have eaten or scattered the remains. So these cool, dry conditions also preserved bone. And in the case of the bone on display, it's soft tissue and its DNA. And the preservation of long fragments of DNA at this site and nowhere else suggests that at the time of the animal's death, the temperatures were very low and probably well below freezing. And the atmosphere also dry enough to prevent any significant decomposition or bacterial activity. And that these conditions then lasted for a period long enough for this organic material to become thoroughly desiccated. Incredibly, analysis of the DNA extracted from this bone helped corroborate that giant ground sloth were only distantly related to today's two-toed sloths, with the two lineages diverging more than 22 million years ago. So, why Mylodon used caves is not conclusively known. Some scientists have proposed that caves were perhaps used as nurseries, since fossils from juvenile sloths have been found there. Others suggest sloths use cave to seek warmth and shelter from the outside environment. Whatever the reason, their presence in caves and the subsequent preservation of material has certainly allowed us to gain a deeper understanding of these ancient animals. With respect to human relationships with Mylodon, current physical evidence shows an overlap in the presence in the area where the skin was found. However, there is currently no evidence such as unambiguous archaeological deposits containing Mylodon remains to allow a confident interpretation of what interaction might have occurred. However, glimpses can be gained through other remarkable treasures, such as this rock shelter painting by the first peoples who resided in the Colombian Amazon. Estimated to have been painted between 11,800 and 12,600 years ago, towards the end of the Ice Age, it depicts humans coexisting with what many consider to be a giant ground sloth. Around 10,200 years before present time, Mylodon became extinct. The exact causes of their extinction are not fully understood, although climate change and its effects on vegetation were almost certainly partly responsible. Despite a lack of complete skeletal material, much is known about the life, appearance, and ecology of Mylodon thanks to these exceptionally preserved remains. And I encourage you to come to the museum and see this remarkable material in person. Thank you.
Thank you so much, Sarah. Fascinating. And I worked on the exhibition and I'm learning more tonight. Thank you so much. Our next speaker, Dr. Robin Wilson, is currently describing new species of marine invertebrates, which are marine segmented worms or polychaetes, from recent explorations of deep sea environments off Eastern Australia. Previously, Robin's research has included environmental studies of Port Phillip Bay and descriptions of new species from voyages on Australia's western continental margin, in Bass Strait and in the Great Australian Bight. Robin is focused on making online identification guides available to all. Robin regularly uses the volume of the Challenger reports that have been sitting on his desk for the past 30 years. So it was an obvious choice when considering which object he might select for this evening's lecture. Over to Robin.
Thanks, Beck, and thanks everyone for tuning in, clicking in, I guess. My name's Robin Wilson. I grew up on Bunwurrung country, and we're in Wurundjeri country tonight. So I'd like to pay my respects to all the traditional owners of the Eastern Kulin Nation and their leaders and elders, past, present, and future. So what are these three little vials of sand doing amongst the company they're keeping in the treasure exhibition? They're among ichthyosaur fossils, finch specimens collected by Darwin, moas, saber-toothed cats. Why are they here? Well, they truly do belong here. They're the first deep sea specimens ever collected on, or they're amongst the collection of first deep sea specimens ever collected on the Challenger expedition of the late 1800s, the first survey of oceans and the birth of marine science. And that's what I would like to talk to you about. So the specimens really are significant in the same way that a vial of moon dust collected by Neil Armstrong is special. Museums are all about the real stuff, being in the room with the real thing. The story really begins, in my humble opinion, with this fabulous map. I love maps, don't we all? Which shows the explorations, invasions of the world's oceans up until the Challenger, because this is a part of the research the scientists did to kick off their voyage and their planning for the expedition. So there's been a hell of a lot of traffic over the world's oceans, but they were like pond beetles skating on the world's oceans. No one knew what was underneath. No one knew what lived there. No one even knew how deep the oceans were. Before I talk about the voyage, let's just give it a moment to think about science in the late 1800s. Evolution was a hot topic, thanks to Darwin and Wallace. They'd published a joint paper in 1858, and Darwin's book was published the following year. Cell biology and inheritance by Gregor Mendel was discovered. Chemistry was a well-developed science. Mendeleev had arranged the elements in the periodic table. In marine science, the azoic hypothesis had been proposed by Edward Forbes, who was a Scot working in the Mediterranean, where actually there are anoxic areas, so he had some reason for saying that. But a Norwegian worker, Michael Sars, and Charles Worville Thompson, who will shortly get the Challenger expedition underway, had already contradicted that finding and shown that animals did exist below for that supposed depth limit. So Thompson, who was a member of the Royal Society, who obviously had a lot of pull in those days, not so much now, were able to release and get refitted from the Navy a vessel, the Challenger, which previously a gunship, so all the guns were removed. It was refitted for marine science. And these are their goal, two of their goals. I've left out a couple about physical oceanography and water chemistry and temperature. The principal object of the proposed expedition is understood to be to investigate the physics and biological conditions of the great ocean basins. In general, you can fit a lot in that, and they did. Another goal was a simple determination of the depth of the ocean at tolerably regular distances throughout the entire voyage is an object of such primary importance that it should be carried out wherever possible, even when circumstances may not admit of anything else. So these are quoted from the narrative of the voyage. Here's the vessel. One of their great innovations, which has been followed by all expeditions since, is to do your very best to keep the messy scientists away from the running of the ship. So the scientists had a deck house on the stern to play with mud in, and there was a dredging deck so that the muddy dredges and other sounding irons, when they came up, didn't interfere with the running of the ship. They had good labs, a zoology lab and a chemistry lab, everything tied down, well set up for surviving, hopefully most of their equipment surviving the rocking ship. But there was another reason for going to sea for such a long time, and with that agenda, submarine communication cables were just becoming a thing. They'd first been tested in the 1840s, and by the time this Challenger was setting out, the first transatlantic cable was already working. And this map, which I didn't know about until I started putting this talk together, shows what an incredible network was already in place in 1901. It was the network of the Eastern Telegraph Company. This was 25 years, say, after the Challenger return, so not long after. They'd already established such an extensive network. So that was one of the several British companies who was great entrepreneurs at the time, capitalised on this wonderful investment by the British government. If you're going to lay a cable, it behooves you to know how deep the water is before you start. So the Challenger filled in lots of gaps. This is where they went, all over the world, taking samples every few days. They went as far south as to have a close call with icebergs. You can read the narrative of the voyage yourself, it's online, and see how dangerous that was for a few days. They crossed the Atlantic several times. They came to Australia, Sydney, Melbourne, and Port Essington, and generally sampled everywhere they went every few days. What did they discover? Well, 362 sampling stations were took. When a ship stops and biologists take samples, it's not a train journey, but nevertheless, those places are called stations. And about 4,400 new species were described. They stumbled across the Mariana Trench, which was good going because that was a very small part of the world's ocean, but they got lucky there. And they were able to sample with a rope, mind you, down to 8,000, a sounding iron down to a little bit over 8,000 metres, which is an incredible achievement. We now know that the deepest part is 10,000 metres, so I sort of always wondered whether the extra 2,000 metres would have defeated them or not. But anyway, they discovered many strange and new deep sea species. But the important thing was that they all fell into familiar groups. They were strange, all right, and they were new species, new genera. They were things which we certainly didn't know about before, but there was no lost world of living fossils, no strange and wonderful things previously thought extinct that would have given extra weight to Darwin's theory of evolution, which is what their secret hope was, I think. In fact, it's sort of poured a bit of cold water over the theory, to be honest, for a little while. We need to salute how very difficult it is to sample the oceans, even now. Ships who go to sea are full of nervous captains. Nervous captains keep us safe at sea, but they're even more nervous when there's a wire hanging over the side of the ship and there's a dredge on this end of that which might get hooked up and the weather might turn bad. In modern ships, it can still take 2 to 4 or more hours to sample the very deep sea. And in the Challenger with ropes, haphazard winch set up was driven by a steam winch over a few drums. It could take 7 hours or more to get a sample back. And during that time, the ship had to somehow try and stay vertical over the rope as much as possible. It's just an incredible achievement that they did. Challenger came to Melbourne. They played a cricket match. They fronted up a scratch team at Melbourne University and challenged the Challenger crew. And there would have been a bit to play for because the Challenger crew were team evolution, whereas the university team would have been led by Frederick McCoy, the then director of this museum. And he was a creationist, didn't believe in evolution. Probably the survival of the fittest prevailed, but we don't know. I don't know who won the game. John Wilde, he came back to Melbourne and his artwork, as you can see there, contributed to the prodromus of the zoology of Victoria. The first publication of this museum on the fauna of this state. They published their report series. They're works of science and they're also works of art. It took 20 years to put them together, publish them, and they're still used now. It was also a collaborative model for modern work. Up until then, these had tended to be one-man shows. But from now on, it was a collaboration, teamwork in marine science. The scientific legacy of the voyage is huge. How many species are in the world's oceans? Well, they wanted to know that question and they found 4,000 odd of them, but we still don't know. In fact, we know less now because DNA evidence is adding to the total all the time. We're discovering cryptic species where we didn't realise they existed. But the other thing they discovered that they concluded was that there's nothing really special, distantly evolutionarily unique in the world's oceans, was still true today. Even the strangest things in the world oceans that we find around the hydrothermal vents, for example, DNA evidence tells us that these are, for all their strangeness, they're closely related to fairly familiar things in shallow water. Evolutionarily speaking, of course, geologically speaking, so their ancestors go back probably 10 to 20 to 30 million years, but not 300 million years or 1.2 million years or anything, 1.2 billion years or anything like that. The truly ancient inhabitants of the deep ocean are microorganisms, but there's no way the Challenger could have learned that. They didn't have molecular tools. There have been lots of successes to the Challenger, but the big one is the Glomar Challenger, which instituted a deep sea drilling program for 40 odd years. In fact, under a different name, it's still going on now. And they discovered all sorts of things about the oceans which would have interested the Challenger. For instance, the Mediterranean Sea, where it all started, the azoic hypothesis, remember? The Glomar Challenger, by drilling in the deep sea and retrieving sediment samples which would preserve the history of the environment, showed that the Mediterranean had died out, if you please. They also found the evidence that gave great support to continental drift, that the ocean basins are young, and in the middle are the youngest of all. So that's where they're spreading from. So these sediment samples really are special. Please come and see them, along with everything else. Thanks for listening.
Thank you so much for bringing the expedition to life for us. Our next speaker tonight, Hayley Webster, is the manager of the museum's library, which includes a rare book collection specialising in natural history. Hayley is the Victorian representative for ALIA Rare Books and Special Collections and the editor of the Bibliographical Society of Australia and New Zealand's newsletter, Broadsheet. And this evening, she will share her insights into the rare books on display in Treasures of the Natural World.
Hi, everyone. I'd also like to acknowledge the traditional owners of the land that I work on, the Wurundjeri and Boonwurrung peoples of the Eastern Kulin Nation, and pay my respects to elders past, present, and emerging. I'm really pleased to be here tonight to discuss my favourite items in the Treasures of the Natural World exhibition. I won't keep you guessing, and there are no surprises here. My favourite items in the exhibition are the books. As a librarian that works with a rare book collection, and as a museum worker that has an avid interest in exhibitions, it always pleases me to see exhibitions that are interdisciplinary and that draw on different types of collection materials to tell stories. To my mind, one of the most special things about working in a museum library is the ability to often look at rare books alongside the collection material that inform them. At Museums Victoria, we have wonderful scientific, cultural and historic collections, and we also have a fantastic library and archive collection to support them. So I'm really pleased to see the exhibition from the Natural History Museum in London make such good use of the variety of collections that they hold. Now on to the books. Historia Naturalis is the oldest book in the Natural History Museum's library. It is not a title that we hold at MV, so I was really excited to see this included. The copy on display was published in Venice in 1469, soon after Johannes Gutenberg popularised the use of movable type in Europe. Within the history of the book, books printed before 1501 are known as incunabula. These books are recognised as an important subgroup of early printed texts within the history of Western printing. Roman natural philosopher Pliny the Elder started compiling the book in 77 AD, and his concept of natural history was very broad. It included zoology, botany, geography, human physiology, metallurgy, and mineralogy. This breadth is common to earlier works of natural history. The disciplines that we know today evolved over time, and the literature that was produced became more specific and focused over time too. An interesting thing to note with respect to this work is that in the early years of European printing, many classical works that had existed in manuscript form were reproduced in print. So this work would certainly have been part of that trend. Before a modern scientific method was developed, natural history relied on repetition of classical sources, common knowledge, and folklore. Later on, the emphasis shifted to observation and experimentation. But prior to this, you might say that science was much more literary or bibliographical, and this work was an important source of information. The next treasure that I will discuss is the impressive Birds of America by John James Audubon. Audubon was a French naturalist born in Haiti, raised in France, and who lived much of his later life in North America. he published one of the most famous bird books, Birds of America. This was envisioned as a grand work of extensive coverage and made for an elite audience. He wanted to capture all of the known bird species in America, and in the end, he illustrated 491 species. Audubon published by subscription, which meant that he courted subscribers to pay him in advance so he could afford the publishing costs. Birds of America was published in 87 parts, each containing images of 5 birds, 1 large, 1 medium, and 3 small species. He gained 151 subscribers and produced around 200 copies of this work. And today we know of only 120 copies that are still in existence. So that makes this a very rare and sought after work. Birds of America was published over a 20-year period between 1827 and 1838, and it was published in a format known as a double elephant folio. This is a huge format, and the volume stands at 1 metre tall. The bound volumes take 2 people to lift them and require bespoke storage boxes and display cases. They are definitely worth seeing in person to get a sense of their scale. To produce books of this size, Audubon had to have special paper commissioned to cover the size. They are the largest bird books ever produced, and the reason for their size is that he aimed to depict the birds at life size. As you will see from the illustrations on the screen, he sometimes had to get creative to stay true to this vision. Audubon was also a naturalist and artist that sought to see the birds in their natural habitat, rather than merely relying on the stuffed specimens available in museums. You can see some examples of illustrations that would have been made from taxidermy specimens on the screen, and you'll see that they are more static, more still, and not anywhere near as lifelike. In contrast, Audubon's work attempts to bring to life the birds by showing them in their natural habitat. He worked in the field to impart life to his subjects, and you will see that they are clearly more dramatic artworks than the illustrations that were based on stuffed specimens, although I must say I appreciate the stuffed specimen illustrations too. Audubon produced the original watercolours that the engraved plates were based on. In fact, he used a variety of artistic techniques to create the original artworks, including watercolour, oil painting, collage, pastels, graphite pencil, and he created highlights using metallic paints or gum arabic. The printed illustrations were made using a technique called aquatint, and they were hand-coloured by a team of up to 50 colourists overseen by Robert Havel. It is estimated that together they produced around 100,000 plates. The volume on display in Treasures is open to a plate depicting the roseate spoonbill, a bird species with very pretty pink feathers. Unfortunately, these gorgeous feathers made this bird a target for plume hunters in the 19th century. In fact, there were many birds that were hunted for their feathers to satisfy a fashion for feathered hats until bird protection laws were introduced. And this happened right around the world, including here in Australia. Regarding the roseate spoonbill, the bird was nearly hunted to extinction, but thankfully the species was saved by bird protection measures spearheaded by the National Audubon Society, named for John James Audubon. Another item on display is very famous for its impact on scientific thinking. That is Darwin's Origin of Species. Darwin's book was the culmination of many years of research, but he was motivated to publish his theory in 1859 after hearing from Alfred Wallace a year earlier, who had also developed a theory of evolution by natural selection. Together, they co-authored an article detailing the theory, which they presented to the Linnean Society of London in 1858. In 1859, when Darwin expanded on his theory in The Origin of Species, there was a huge demand for the work, and the first edition sold out almost immediately. The first edition was published on the 24th of November, 1859, and copies of this work arrived in Australia by early 1860, not too long after it was released. However, at the National Museum of Victoria, our predecessor institution, the director, Frederick McCoy, was not receptive to Darwinian theory, as we've already heard, and he did not acquire a copy of the work for the museum. In fact, by the time the museum was looking to purchase an early edition of the work, this time for its historical rather than scientific significance, it was 1999, and we were only able to purchase a second edition, which, as you might imagine, is a bit of a sore spot for me. The fact that Frederick McCoy did not acquire a copy of Origin of Species did not stop him participating in the public debates about evolution. He infamously obtained gorilla specimens to display in the museum in order to suggest how absurd the notion of any relationship between humans and apes was. Evolutionary theory was debated throughout the 1860s and 1870s and was certainly not unanimously well received. However, towards the end of the century, it had become more widely accepted and biology was being taught in Australian universities. I hope this has given you a taste of what you might see in the exhibition and a little bit of an insight into the world of rare books in a natural history museum. Thank you.
And that brings us to the end of the night. Thank you so much for joining us.